So coming to congenital malformations, they are a pretty common occurrences. They occur at approximately 1 in 1,000 of births, if you take from birth registry. And half of them may be detected after birth, because many times we may not be having the facilities to detect them early. But major ones are easily detected, and now we are proceeding more towards MTP. So quarter of the patients will be those who will be needing MTP sometimes. So we need to identify them antenatally, and we need to identify them postnatally. So antenatally, ultrasound is the first modality to go. If the ultrasound is inconclusive, then we go for a MRI. And postnatally, we need a MRI, of course, and at a high resolution and at a 3 Tesla scanner. So one thing which is you can say about congenital malformation is that there are at least 2,000 described malformations, and we have got so much huge numbers. Then probably the biggest job is to group them together so that we can place them, what kind of malformation we are looking at. So my job today would be to give you an overview how to place this malformation, so that once you occur them, then you can recognize which malformation it is this. So multiple attempts have been done to classify congenital malformation based on, say, etiology. But the problem is it can be genetics. A lot of them are due to genetic factors, but there may be epigenetic and environmental factors. So it will not work for some of them. If you are looking for morphology and structures involved, most of the earlier researchers were doing like this. But the problem is no two malformations are alike. So going by morphology alone, also you will not be able to classify many of them. If you are going by the embryological, the developmental stages which are being involved, then the problem is, if you are looking at the stage, when the one single gene or single protein is actually occurring at multiple developmental stages. So placing them into single developmental stage also doesn't work. If you are looking for genes, again, there may be one gene may be involved at multiple parts. So this may be causing multiple types of malformation, or there may be multiple genes which may be causing the same malformation. And we know that underlying problem is, of course, a molecular pathway. So these are a lot of problems. But the thing is that even though we have identified the problem, there may be many different phenotypes, and that the different parts of the brain may show different appearances due to somatic mosaicism. There may be regional differences. So what I'm talking about, that molecular pathway. So say take very small pathway, which is hypothetical. There's a gene and a B gene, and they both interact to give you the D product. So the problem may be with A or B. But what happens is many times the B gene is being also influenced by a C gene. So now, and you can remember that most of the pathways which are involved in brain formation are multiple A genes. And so you can see the huge numbers of permutation and combination which can occur. And this can result, so what's different permutation represented into phenotype A, phenotype B, or there may be another phenotype which is having multiple abnormalities. So what the approach today we will be following will be, we will look at the developmental stage, we will look at the particular process which is involved in that stage, and the structures involved at that time. So by a combination of these three, we would like to place and identify the malformation. So we know that neurogenesis, the first part will be the formation of the neural tissue. So neurogenesis, it has got two parts. First 4 weeks are for dorsal induction, and the next 4 weeks are for ventral induction. So these all timelines are very simplified from my side. If you look at the books, then you may find 1 or 2 weeks change maybe there. So what I've done is 8 weeks of neurogenesis, 8 weeks of neural proliferation, differentiation, and histogenesis, and 8 weeks of neural migration. You can remember for your purpose. It is not exactly so much state jacketed. And then you from 16 to 24, after 24 weeks till 2 years of age, both post-migratory organization is happening and myelination is happening. Many of the processes may be not due to primary involvement. They may be due to secondary insults like hemorrhage and ischemia. And of course, many of the cases will not have any known cause. So malformations of cortical development basically belong to the group B, C, and D. And this is also part of my talk today. So starting with the disorders of dorsal induction. So when you're talking about induction, what does that mean? That something else sitting somewhere else is inducing. It is not in contact, it is inducing, and this happens to the chemical signals and transcription factors. So when we are dealing with dorsal induction,